Okay, let's start with the first team. Thank you, everyone. Welcome to this is the room number 5 for the judging session for Design League 2023. And it is for the environment and biodiversity group. So today we will have 4 teams in this session. I will general instructions. I will start sharing the promotional video for the team, and then we will have 20 to 22 minutes for the judges to ask any questions they might. So we will start today with the first team. Team members, please, if you have any more team members in the waiting room waiting to log in, please instruct them to add their team name to their Zoom name so I can identify them and then let them in. I will start sharing your promotional video now.
Porque nadie había comentado de su gravedad.
No creímos que fuera tan importante. Tumberjalata o ojo de poeta es una especie nativa del oriente africano.
Al grano, ¿qué está haciendo en Colombia?
Solo mírala, es hermosa. Fue introducida en América en el siglo XIX como planta ornamental. Mira sus bonitas flores naranjas.
¿Acaso están de su lado? Cubre bosques, devasta cultivos, compite ferozmente por recursos con las plantas nativas. ¿Qué la hace tan exitosa?
Una planta increíble, puede autopolinizarse. Su fruto revienta esparciendo sus semillas de 2 a 3 metros. Se reproduce con esquejes. Hay casi 1500 semillas por metro cuadrado, con germinación en 72 horas. Y las asociaciones micorrízicas, el clima y los insectos son sus aliados.
Distribución.
En Colombia se registra en todo el territorio. Nos preocupa el altiplano del oriente antioqueño, con observaciones entre los 1500 y los 2900 metros sobre el nivel del mar.
Solo en el oriente y en zonas tan altas. No es una planta nativa de zonas costeras.
Eso también nos preocupa. Se ha observado en ciudades más bajas como Medellín y Bello.
Quiera todos fuera ya, solo requiero del personal científico. Aliados.
La alcaldía de Envigado y Cornares están trabajando sobre el tema. Tenemos aliados con el grupo de investigación de la Universidad Católica de Oriente, con los campesinos afectados, con escuelas y con la comunidad.
¿Y por qué no hemos podido con ella?
Una erradicación manual resulta ineficiente, el control químico peligroso y por control biológico se tiene desconocimiento sobre la planta.
Esta vez perdimos, ¿verdad?
La biología sintética nos podría ayudar. Le presento Poetizar.
Los RNAs de interferencia son una herramienta de silenciamiento genético de organismos eucariotas como Tumberjalata, las cuales cuentan en su maquinaria celular con una serie de complejos proteicos que facilitan el ancla de dicho RNA a un RNA mensajero complementario, reprimiendo su traducción. Nuestra estrategia consta de la producción de escala de los RNAs de N. coli con el poder de silenciar la expresión de un gen de relevancia metabólica en ojo de poeta, logrando su aniquilación. Usaremos un mecanismo de expresión que facilite su distribución en nanopartículas que permitan su absorción en las células vegetales y conservar la integridad de los RNAs, garantizando selectividad, efectividad e inocuidad.
Thank you. Now we have 20 minutes for the judges to ask questions. If you have any questions, please raise your hand so we can start.
Hey, sorry, I think there's been a mistake because that is in our video. I think there's been a mistake. We are team HS2 1L.
Yes. Okay. I'm sorry, I will replay your video.
Yeah, it's fine. Thank you.
Nuevo León, a state located in the north of Mexico, is currently known for its economic growth and value to the country's industrial processes. But even though the life quality is going up with the economy, The air quality keeps getting worse and worse each day. Polycyclic aromatic hydrocarbons are a big part of this atmospheric pollution problem that can be seen around the whole metropolitan area of Monterrey. This class of more than 100 different compounds with almost the same characteristics are produced by the incomplete combustion of organic matter. They are formed by two or more benzene rings attached together, making it hard for the body of living things to degrade. According to some researchers from our city, the levels of creatinine were up to 0.76 units. That means more PAHs in the system, in which those levels were comparable to an adult smoker. The toxicological effects PAHs have on the body from the prolonged exposure to them go from irritation to possible cancerous cell damage, and the only way of protecting from them was buying expensive filters capable of filtering these small particles.
That's why our team has come to action and designed a way to detect and degrade this compound to protect the life and environment of the place we live in. This is by implementing recombinant catabolic proteins and a signal protein with GFP to detect and minimize the presence of these compounds. By the regulated production of three catabolic proteins in charge of PAHs degradation, we are aimed to make our bacteria act as a biofilter. On the other hand, by adapting a natural detection system coming from bacteria able to metabolize such compounds. We are aiming to develop a detection system through the production of GFP protein, signaling the presence of these hazardous gases in the environment. The signaling and degradation that we will be getting could help in the risk assessment of industrial working areas to help reduce the overexposed workers and families living nearby.
Thank you. Okay, we can start with the questions now. If you have any questions, please raise your hand. Yes, Telma.
Hello, team. Congratulations first for a very good job. And my question is a little bit about the market research you made. I know you're trying to do like an air purifier, but what about the mechanic ones, like Dyson, like all the ones that are already sold? I know they don't have biotechnology, but I think they are way cheaper. So how is your product going to be competitive in that way against the ones that are already there in the market?
Okay, I'll be happy to answer that question. Okay, so yes, we investigate other filters that exist right now in the market, but since this is a type of compound that is not really talked about, there aren't very filters that are for this specifically. And the thing with these gases is that they are not really well investigated. They haven't been, and the effects that they have on the body are bad from the prolonged exposure to these type of compounds. So what we wanted to do with this was to detect, and that was the most important part of our project, because if you cannot detect that you're living with these hazardous gases, then how come are you able to buy a filter to degrade them? So with our project, we are aiming to first detect that, yes, we have the gas in the place that we live in, and then attack the problem with the same biofilter. So that's the first advantage that we have. And the second one is that they are not very cheap. We investigate, and the pricing of the filters that we know can like trap these gases is like in a range that's similar to what we think our prototype is going to be if we were to make it a real product. So that's another advantage. And the other thing is that all of these filters that are mechanic only trap the gas but not degrade it. They are not capable of doing such thing. So we think that we have really good advantage over all these type of filters. And also our biofilter is capable to detect. So that's another great thing about it.
Okay, thank you.
Thank you. We have one question from Lucas.
Yes, hello guys. Congratulations on your great work. I love it. I want to know more about your gene expression. So why is too important in your project the vanilla acid? So I want to know more about that, the promoter that you use. To tell me more about that, please.
I think my team members are having trouble getting in the session. So I think I could answer this one too. So we use an induction system that is by vanillic acid. This is because we think that producing three enzymes could be really stressful for bacteria. So we were wanting a system that could make it possible to produce the proteins that we wanted when we wanted to. So with this system, we can make it possible only when vanillic acid is in contact with our bacteria. So in that way, we can control how it's producing. And I think that is pretty important because with the biofilter, we want to be able to make a really good understanding of how are the bacteria working and how the enzymes are working. So with the system, I think it's a pretty great option that we managed to have, because if you see, we have our promoter and it's being repressed by a factor van air, and then vanillic acid, what is making it's to make it possible that this promoter starts producing all of these enzymes.
Thank you.
Nice, Daniela. Thank you.
We have a question from Margareta.
Yes. Thank you so much for contributing to this competition. I really like the focus of your project, and I think you work on a really important problem too. So having a biosensor or a biofilter that does both detection and degradation, do you think both those function needs to be in the same biofilter at the same time? Or it's very ambitious. Which, if you have to choose, is the most important?
I don't know if Andres want to answer this question.
I could rephrase it.
No, it's fine. I get it, but I want him to answer if he wants to.
Sorry, you can respond to this question, Daniel.
Okay, I will. So as a team, we had that too. We thought, why is it important to detect and also degrade? So as I said before, the thing with these compounds is the investigation that we have. And we thought that making the two things was ambitious, yes, but also it was necessary, because with this, we are bringing that to people that need it. Since the polycyclic aromatic hydrocarbons are produced by incomplete combustion, in our state specifically, we have a lot of industry going on. So there's a lot of people who are breathing these gases and that are getting into their body and are having, well, difficulty breathing, or they have irritation, or they have possible cancer cell damage, which is a really important thing because that can lead to cancer. So we thought that these people needed a product that was efficient and also really helpful us in many ways as possible. So we thought that we needed to do as many things and as many good things in the project, and that's why we implemented both things. But we do think that detecting them was the most important step, because the ways that we can detect these gases are chemical process that are really more complex than just with the GFP protein. So the process, when we made it genetically, it was very much easier than taking samples, getting them to a lab, then making chemical processes to them. That's an expensive process that not a lot of industries are willing to do. So we are just trying to make it a lot simpler with our project for the people that really need it and the industries that really need it and want to do something positive for the health and the environment as well.
I like that answer. Thank you.
Thank you. If the judges have any more questions, you can raise your hand. Yes, Lucas.
Thank you, Patricia. Yes, I see that you are working in bioart, so I love it. Nice work. So I want to know better, more about that project. of bioart. So I would like to know how you did that, those experiments in your school, because you are a team from a school, uh, right? Okay, and I want to know more about that process. I very interesting for that.
Mm, okay, so in our high school, we did a science week. We thought that it would be pretty interesting to have that kind of attention because not a lot of people knew about our team. They thought, oh, an investigation team, isn't that like from universities and stuff? And we thought, well, why not do it in our high school? And our mentor told us, well, we can make like this initiative and we can show it to the principal. So we planned it and we did it. And we wanted to have a focus on biology and STEM and sciences, but more specifically in biotechnology and synthetic biology. So we knew a lot of people from the medium because in our high school, it's a high school that's implemented to a university. So we knew people from the career of biotechnology. So we talked to them and we told them, do you know someone that can do these projects with us? And they teach us a lot about biosafety. They're actually from a team that's in Design Leaks as well. We collaborate with them. They're called um uh YNL team. Yes, they are from FCV. That's what they call. So we collaborated with them, and they told us a lot about the type of things that we could do to that were also saved. So we work with a group of like about 30 students. We had them responding to a Google Forms where they put their names in the classrooms, and then after classes, they'd come to the laboratories from our high school, and obviously our high school managed to give us all the things that were needed, and we had all of this support, and we did that with the help of our mentor. He knew about biotechnology, so he helped us. And then the bio art activity specifically was a pretty interesting one because what we realized while making it and being a staff in there was that a lot of people didn't knew that they could be capable of doing all of this stuff with their age because we are high school students. So they thought they was really fascinated. And when they saw the results, they were fascinated as well. And not every single piece of art was like really, really professional, but they had fun doing it. And I think that's what good. At the end of the week, we realized a lot of people were interested in science and they didn't thought that it was boring. So that's a good thing because we managed to get to those students that didn't knew about the team, and now they know, and now they know about biotechnology, and they're excited and happy to know about it.
That was awesome, Daniela. Thank you.
Thank you. We have one more question from Thelma.
Hello. Coming back to your project, I see you were trying to also biodegrade these important compounds, the polycyclic aromatic hydrocarbons you found. So what are the products of the degradation? Because sometimes when you break a molecule, it becomes like a smaller, worse particle that may be more pollutant than the original ones. So what are the products after the degradation that you found with your idea of the degradation pathway?
Let me just, can I have a second to check because I have that on my notes and things.
Yes, of course.
Okay, so while working with this type of compounds, we knew that there was a possibility that there might be products that are toxic, and well, worse, because that's what happened with most pathways. Well, not most, but with some. So what we tried to do was to think as logically as we could, and we thought about different pathways that were with these compounds. So we investigated, and there's a lot of bacteria that could do the degradation for different groups of polycyclic aromatic hydrocarbons. There are certain pathways that they can have. They can have a meta pathway. They can have high. I'm trying to introduce because I learned them in Spanish, but they have different names. The pathways that we investigated. So we know that the products aren't toxic because with these bacteria, they degrade them. And with in case of the protocateguate, that's one of the enzymes. What it does to the gases is to effectively get them to the TCA cycle so they can be converted into energy. So we know that these compounds aren't being converted into worse compounds because they at the end what they are doing while degrading is to get the carbon and transform it for something good to the bacteria. That's why they do it. They do it because they want the energy that comes with the compound. So that's how we know that we aren't like making it worse. But there are certain different products that we have because there are 7 different compounds that we have.
Okay, thank you.
Thank you. We have time for one more question if one of the judges have any questions for the team. Yes, Margarita, go ahead.
A quick one then. Are you aware that plants actually can accumulate these hydrocarbons, these toxic compounds?
That what, sorry?
That plants actually can pick up. You want to
Yes.
comment on that?
Yes, I like to. While investigated, we realized that these compounds are lipophilic. I think that's how it's said in English. So they have this characteristic that they like to be on lipids and well, all of these grasas. I don't know how to say it in English, sorry. But they like this characteristic. That's why they are more likely to be in bodies or plants. So this is damaging for them because at the end they're toxic. This is the environmental factor that we wanted to attend as well. Not only the health of humans, but only the health of animals and plants, because having them can make animals infertile, which is something that's pretty important to us too. So we are attending 3 different things: the health, the responsibility from the industries that are emitting these gases, and also the environmental factors that are being taken over by these gases.
Thank you.
You're welcome.
If we don't have any more questions, we have 1 more minute before we finish the session. Yeah, okay. So if we don't have any more questions, so I will ask the students for this session to please leave the room so we can allow the students from the next session to log in.
Thank you.
Thank you.
Thank you.
Thank
Goodbye.
you.
Thank you. Congratulations. Keep going ahead. Ciao, ciao.
Okay, and now I will let the students from the next team to join. Welcome, students from IGEM Evolutech PA. General instructions, I will be sharing your promotional video, and after we finish, you will have from 20 to 22 minutes to reply from questions from the judges. If you have any more team members that are in the waiting room trying to log in, please remind them that they need to add your team name to their Zoom name so we can identify them and then allow them to log in into the session, please. I will start sharing your promotional video now.
In recent years, the problem regarding soil erosion in Panama, specifically in La Villa River Basin, has experienced a severe drought, desertification, and erosion, negatively affecting biodiversity and other aspects of nature. That's why we created Ground Saviors, a pill that is soluble in water and it is made up of algae that has been genetically engineered to produce cytophores. With this, we battle soil degradation by reducing the amount of kalinitic clays found in soil.
To produce siderophores, which are organic compounds that bind to metal ions present in clay, such as iron and aluminum, and solubilize them. Our system will have two gene overexpression circuits. The main enzyme that uses NRPS acts in collaboration with the adenylation A modules to recognize and activate the corresponding amino acids. The second is to generate the necessary amino acids for our final circuit that will have the CoA-A, CoA-D, ACCD genes and the amino acid tRNA synthetases gene. In the end, through biosafety process, an inducible cell-state circuit will be made.
We thoroughly studied the strains and potential project impacts in various areas. Our team is now working on a safety assessment, especially regarding the project's release and environmental and business risks once it gets public approval. Our project seeks to raise awareness among young people about soil erosion in Panama, supporting the CDGs 2, 6, and 15. Technology and ethics are central to the development of biotechnology, a valuable tool for addressing environmental challenges and a promising solution to soil erosion. Ground Savior, the transformation that turns erosion into regeneration, creating healthy grounds.
We can start with the questions from the judges now. If you have any questions, could you please raise your hand? Yes, Lucas.
Hello guys, congratulations for your works. Yes, I want to know more about your chassis. Why do you utilize the algae as a chassis, and specific why you use that species of algae?
Well, I want to respond first. Well, we choose Spirulina platensis and Cyanecosystis sp because they are the algae with better storage when it comes to the genetic circuits, since they are very heavy, and that is also why we divide them into two algae. And we choose Cyanecosystis sp2 because it was the best option for our NRPS gene, because it has a better gene expression in that algae.
Okay, thank you, Ricardo.
Thank you. We have one question from Margarita.
Yes, hello. Happy to look at your contribution. So I have a question. Could you say something about the soil problem? I'm from the northern part of Europe, and I don't think we have kaolin soil. Could you say what is the problem with the kaolin soil that you target in this project?
Okay, hi, my name is Alejandra. So here in the region that we live in is called Azuero, and there is a very huge problem with the erosion because of the bad practices in our culture and etc. And also the contamination and pollution of the La Villa River, that is the one that provides water to the whole region. So those factors combined, well, they worsen the condition of the soil, therefore having consequences in the crops and the harvesting process. So we wanted to focus on this because agriculture is a very primary activity here in the region, and we wanted to improve those activities.
Thank you.
Thank you. Telma, go ahead.
Hello, people. Congratulations. Very good work. And I would like to ask, why a soluble pill and not only like a powder? I imagine that this product is for the farmers that are trying to create their new crops. So why is it more useful to have a pill instead of just a powder that you can put in water? Why the presentation?
Firstly, we are using translators between the members. So, is there any translator that can translate the question, please?
Okay, so
Hello, I'm one of the translators. I want you to please can repeat the question. I didn't listen very well.
Yes. You are creating a pill that is soluble in water. So my question is, why not only like the powder or any other in any other way to put your product into the soil? I think the farmers, for example, may be the same for them to use a pill than to use a powder. So why exactly do you want to have this presentation as a pill?
Okay, now I'm going to translate it to the team members. Ok, nuestra jurada aquí presente nos acaba de decir que estamos utilizando una pastilla, que por qué no utilizamos otro elemento como polvos, otro tipo de elemento.
Bueno, nosotros, para que puedas traducirlo, lo que nosotros estábamos intentando hacer era no solo que fuera posible sea más fácil de manejar en términos de adaptabilidad, sino porque nosotros estábamos diseñando un sistema de riego por goteo. ¿Alguien puede traducirlo? Alejandra.
Okay, so the team focused on a soluble pill because of the adaptability that this has. Also because of the development of a, give me a second, sorry. Yeah, a watering system. We also developed a watering system that Design of Experiment could talk more about that.
Fine, thank you.
Well, I'm gonna explain a little bit more about this el sistema de riego. Estoy intentando.
Watering system.
Irrigation system.
What?
Irrigation system.
Okay, okay. Que es un sistema por goteo. No sé cómo pueden traducir eso.
Irrigation system.
No,
Drop by
pero
drop.
por goteo.
Drop by drop.
Drop by
Goteo,
drop.
I think is drip system, if I'm not wrong.
Y bueno, este sistema nos ayuda mucho para poder distribuir de manera equitativa y distribuir también de manera eficiente a las diferentes plantas que haya en los cultivos. Alejandra.
So the irrigation system by drop allows us to distribute it more equitable to the crops and the harvest and the soil in general.
Además que debido a que cada diferente planta necesita un tipo de cuidado diferente, con este tipo de sistema que está basado en microtubos y sistema de botones para dar más o menos agua con este tipo de solución que va a estar dentro del agua que se le va a regar para así evitar lo que es todo lo que podría surtir mal por la sobrecultura, no sé cómo se podría decir, o el desgaste de los suelos.
So basically, what my partner is saying is that every plant has a characteristic care. So with the irrigation drop system, we can monitor and control the drop and the watering of each crop more easily and more effective.
Eso es todo.
Thank you. Lucas?
Yes, thank you, Jesus, Alejandra. Now I understood better the implementation. Nice. I want to know about the genetic circuits. So you choose 5 or 6 genes, main genes. So I want to know more about that genes. And if you want to choose 1 or 2 genes or protein, which one you wanna choose and why?
Bien, yo voy a responder. Denise, you can translate. Ok, nuestros genes principales, pues la verdad es que sin alguno de ellos no funcionaría completamente. Pero, por ejemplo, nosotros estamos abordando 2 funciones. La primera, que es la de la producción de humus, en el que utilizamos el gen CEL5A para degradar la celulosa. Y el segundo, que es la producción de sideroforos, que utilizamos los otros 5 genes para producir los aminoácidos necesarios para esa producción de sideroforos. Denise.
Okay. We're like using 2 functions. The first, which is with a humus, if I'm not wrong, production to decrease cellulose, and the second, which we are using the other 5 for the production of, what was the name, Howard, again?
Siderophores.
Siderophores.
Sí, entonces, pues si tuviéramos que elegir entre uno o el otro, pues podríamos dividir las funciones y elegir el de la producción de humus, que también aporta mucho al suelo, ya que es solamente el gen de la degradación de la celulosa, y entonces el gen de la degradación de las arcillas cauliníticas, que son los sideroforos, pues entonces, pues, se podrían dividir de esa manera.
Perfect.
I have to translate, right?
Yes, I
Okay.
think it's better.
Okay. He said that that it's better to divide 2 of them. Can you repeat Howard again, please?
Y sí, que es mejor, si tendríamos que elegirlos, podríamos dividir en las 2 funciones, la producción de humus
He said that we can divide the production of the 2, one for the production of the humus. Continue, Howard.
Y la producción de sideroforos, ya que la producción
And
de
the production
sideroforos
of
tiene,
cider force.
ya que esta tiene más genes que tratar. que la de
Okay, because that one has more genes to manage than the other one with the humus.
Okay, thank you.
Thank you. Do you have any more questions from the judges? Yes, Margareta, go ahead.
Thank you. So I'm curious about the human practice part. Which part of the human practices do you think was most successful and why?
I think my teammate Dennis can answer this question.
Can you repeat the question, please?
Yes, I'm curious about the human practices that you performed in the team. Which part do you think was most successful and why was it successful?
I would say the interviews to professionals because we learned a lot of things through them because they teach us about the grounds, about how to preserve them, about the plants. And we think that that helped us to make the team know more about our main topic, that is the grounds and how to save them.
Thank you.
Yeah.
Hey, I wanted to add to what my partner says. We had, yes, the interview with the professionals that help us understand more about the problem of erosion here in Azuero, but not only those interviews, but also we did interviews to the community to see if they understood how big this problem was. And we were made aware with the results of our interviews that most people here in Azuero have no knowledge of how huge this problem is and how can affect all the province in the future. So I think that has a huge impact too about the human practices.
Thank you.
Okay. Do we have any more questions from the judges? Yes,
Yes,
Margareta. Oh, sorry.
Okay. No, if
Margareta, and then you
Margareta
go ahead.
wants, yes.
Go ahead, Lucas.
Okay, thank you. So I want to know, I'm curious about your bio art project. I see that you draw something. My question is about why you choose that type of art and not another type, and why you do that?
Okay, I was one of the people who draw, and I made the draw of the tree that looks like an DNA tree with colorful leaves. If you notice, I use the colors of biotechnology. I use blue, I use green, red, orange, and all the colors that represents biotechnology based on the different uses that they had. I also add some ground in the under the tree to symbolize our project that is made with our main topic, that is grounds. And I also made like a letter in a cute letters on the side, so you can see that we are very, very happy to be in this competition and how we are being working hard to get in here.
Thank you, Dennis. Congratulations.
I also want to answer that because I also was one of who creates the art. For example, I make much of Minecraft and other things because for me, for example, for that games or for example, the art that I make like for some students with the scientist look with the little plant on the top. I make, for example, that type of drawing because it's represent how we need to care about the earth, the ground, and how if we make our contribution or our help to save our planet, to save our ground, because it's so essential. In the ground, it's making all our life, all the plants. So if we don't care about it, we can't survive in this planet. So that's why I make that type of drawings where it's much people taking care of plants, or like the type of pixels in Minecraft, because this type of games care about the planet, about the animals. So that's why we and I create that type of drawings.
Yeah, so basically, all of our artworks was based on biotechnology and creativity, referring out to our project that is about the grounds.
Thank you. We
Perfect, thank
have, sorry, we have another question from Margaret.
Well, when it comes to biosafety, is your future product going to be safe to put in the environment? What is your short answer to that?
Okay, so first of all, both of the strains that we selected are biosafety level 1, so they are not hazardous to any organism. And also, we developed a 3-step biosafety method. That is one that confined trials that we will use samples of soils that are eroded to put into development the project before the environmental release, which is the step number 2, and where we will monitor the project and the development. And then is the market release. And when we know that it's safe, when we have the approval of the national entities like the Panama Biosafety Project, and we are sure that we adhere to the guidelines from the Cartagena Protocol.
Thank you.
Thank you. Thank you. Do we have any more questions from the judges? If we don't, then no. Okay. No. Okay, perfect. So that ends this session. Thank you to all the team members from iGEM Evolutech PA. I will ask you to please leave the room now. And yes, thank you everyone.
Bye.
Ciao,
Thank you to have
Okay,
Gracias.
thank you for your time.
Have a nice
Thank
Have a
you.
nice
Good
day.
luck.
We're halfway through the session, so we can take 3 minutes if you want to go get water, coffee, or something. Great.
Yes, it's important.
Short break.
Gracias,
I'll be
We'll be
back.
back in 3 minutes.
Okay, gracias.
Okay, everyone's back, I think. I will start letting the next team in. Welcome team members from IGM Ecosoil. If you have any more members waiting to come in the waiting room, please just remind them that they need to put the name of your team so we can identify them and let them in, please. I will give you 1 minute. They're joining. So general instructions, I will be playing your promotional video for everyone. And after we finish, you have between 20 to 22 minutes to answer questions from the judges. So I will start playing your promotional video now, and while we wait for the rest of your team members to join.
Costa Rica es el principal exportador de piña a nivel mundial, pero hay un factor que es contraproducente. En el país se hace uso del mancozeb, un fungicida bastante reconocido entre los agroquímicos que se utilizan en las plantaciones. Este principalmente se degrada en etilentiurea, o bien se le conoce como ETU. El ETU puede permanecer en suelos arcillosos, como los utilizados en los cultivos de piña. Además de esto, en el agua y en el fruto como tal por prolongados lapsos de tiempo. Este contaminante puede perjudicar a los consumidores de piña, pero principalmente a las poblaciones aledañas a las plantaciones, debido a que no hay un correcto tratamiento del suelo. Entre las consecuencias con respecto a la salud de la vida humana se encuentran daños en el sistema digestivo, glándula tiroides y además es cancerígeno. Por otro lado, en cuestión del terreno, este afecta a la absorción de los nutrientes.
Por esto, hemos buscado una forma de biorremediar el residuo contaminante. Gracias a las herramientas de biología sintética, introduciremos en una E. coli las instrucciones para producir una enzima llamada manganeso peroxidasa, la cual es capaz de degradar al ETU. Al degradarse el contaminante, este se convertirá en lentilentiamina, que a su vez genera otros productos biodegradables potencialmente beneficiosos tanto para el suelo como para las plantas. De esta manera, lograremos una disminución en las afectaciones en la salud de los productores a causa del ETU, permitiendo un desarrollo más sostenible del cultivo de piña en nuestro país.
Okay, we can start with the questions now. So if the judges have any questions, please raise your hand. We have 20 minutes. Yes, Margarita, go ahead.
So I will start by congratulating you on your project. Very interesting topic chosen, and very many different important perspectives in the project. So I want to start by asking about the toxic compound ETU. How long does it stay? You say somewhere that it stays a long time, so maybe you can comment. How stable is the compound in, for example, fruit?
Okay, in general, the ETU still remains for about 2 or 3 weeks in the soil, in the plant, in whatever of the parts of the plant, like the leaves or the fruit. So that's it.
Thank you. Or maybe just a follow-up question, because there's some information that the ETU molecule degrades in 3 days. So do you have a reference, or is it different in different parts of the world or different analysis?
We have a reference of a study of the World Health Organization about the uses of pesticides of different types in Costa Rica. So the principle of the class of fungicide, it's mancozeb. That it's the mancozeb degrade about 2 days, but generate the ETU and remains 2 weeks. So there is a study of.
Thank you.
Thank you. Thelma, please.
Hello, team. Congratulations. Very good work. And my question is, if mancozeb is like so bad for health, what is the regulation about it? What if at some point mancozeb gets like canceled or absolutely banned? Then what would you do with your product? So first of all, what are the current regulations, and why do you think it is still being used if it is so bad for health?
Well, actually, it's because Costa Rica is very permissive with these topics. So this is the reason, personally, because in other countries it's not allowed to use it. So also in Costa Rica, we have like no other method to. Actually, it's because it's needed to use also. So given that we do this solution, so we can use mancozeb, but in a secure way.
Thank you. We have one question from Lucas.
Yes, very good, guys. Very nice work. I had a question. Is synthetic biology the best option to treat the mancozeb problem? Are they bioremediating? Did you think about the possibility of making a product similar to mancozeb with synthetic biology, but better than mancozeb? So I want to know more about the problem and the relation with synthetic biology.
Okay, first of all, a project consists in a solution of a local problem of a country, right? So when we create this project, we thought like we need to solve this problem, and we have tools, for example, biology synthetic. So this is why we create this project using the tools that we have. But also, I think that we can create like another alternative if I'm
okay with the question. I mean, we can use another alternatives to create, for example, agrochemicals as Mancozeb. But I think that as a solution to the problem, the idea to create a prayer, for example, EcoSoil to degrade Mancozeb is like a good alternative using the biology synthetical that is not common use and in our country, and is like something new and that can like fix problems like this.
Very good. Thank you, Alanis.
Yes, Telma.
Thank you. So correct me if I am wrong, but your idea is to have like a gel with the degrading protein directly to put that into the soil. Is that your correct idea of implementation for your design?
Yes, we want to extract the enzyme from the bacteria, and then we make the emulsion because it is easy to apply.
And what about you say the ETU is also like in the fruit itself? Do you need to clean that with your solution, or what would happen with the ETU directly on the fruit, not only in the soil?
Okay, well, first of all, the use of Mancozeb to protect the plant from fungus infections, it's important to tener en cuenta, I don't know how to say, because we can use EcoSoil to eliminate Mancozeb before Mancozeb destroys or protect the fungus infection. So the idea of the application of the enzymatic emulsion is that first the producers, the farmers uses the Mancozeb, uses the pesticide, and days later, when the Mancozeb already killed the fungus or protect the plant in question, they will use EcoSoil, so they can clean the plant. So but before the EcoSoil stands before the Mancozeb in all of the production cycle. In the first when the seeds are growing, when the fruits are maturating. So I think that I answer the question.
Yes, thank you.
Thank you. Do we have, yes, Margareta, go ahead.
So I think it's good to distribute an enzyme rather than the bacteria. But do you have any information on the stability of the protein that you want to release, the enzyme? Do you think it is possible to distribute to stably get rid of the toxic compound?
Of course. Actually, the manganese peroxidase, the enzyme that are we talking, naturally, it's produced by the fungus Phanerodontia chrysosporium. This fungus eats vegetal material like leaves and roots. It's a basidiomycete fungus. So the problem is that if we use the manganese peroxidase from this fungus, we are going to destroy also with the pineapples or the plantation. So when we obtain the sequence of the manganese peroxidase and optimize it for E. coli, we notice that if the manganese peroxidate is produced by a microorganism like bacteria, it needs oxygen peroxide to degrade the plant. So there is no risk that our protein it will damage the plants, and the structure and the stability of the protein is the same that the Phanerodontia species produce. So there is no inconvenience of the stability. And we have a study that reveals the need of oxygen peroxide, and in manganese peroxide produced by B. subtilis, and E. coli also.
Thank you.
Well, also in addition, there are some factors that are experimental. For example, the pH of the soil. So there are factors that we can determine now. So it will be in the next phase.
Thank you. We have one other question from Lucas, please.
Thank you. Yes, about the computational prediction. I thought that you did docking, so I would like to know what is the main information that you extract from that test. Like, what is the result that is most important in your docking test?
Okay. We use principally two programs or two pages for the docking. One is HDoc page, and other is SwissDoc docking. And in both programs, we obtain that the ligand is attracted to the binding site of the protein, and this is the most important result in both pages because it confirms theoretically that the ligand will binding to the binding site of the protein.
Thank you, Angulo.
Thank you. Yes, Telma.
You mentioned at some point that some of the products of the degradation was ethylenediamide and also glycine. But which other compounds come out of the degradation, and are they safe? Because sometimes smaller molecules tend to be like more toxic. So what are the products and if they are safer?
Okay. We obtained that the most predictable product is ethylenediamine, and we know that this product will not damage the soil and the plants based in articles that say that first, this compound is beneficial to sobrecarga excess of zinc in the soil. That in fact the zinc is other problem of the mancozeb. This in one point. In other point, it is compound ethylenediamine is biodegradable in glycine, and glycine is biodegradable in other substances that are beneficial to the plant and to the soil. And in case that in case that we obtain other products, it in articles mentioned that is probably to obtain chelators from the degradation of ETU. And chelators are also biodegradable in the soil and not represent a damage to the environment.
Thank you. We have one question from Lucas.
Thank you, Patty. Yes, a quick question. I see that your mentor is here. So I want to know how was the relationship with the mentors and team members to construct your project? Because it's a very important relation.
Bueno, first of all, we already know our advisors, our mentors, after IGEM Design League. We participate in a competition of synthetic biology made by IGEM Panama and the Bridge Biofoundry a company in Panama too, like a bootcamp and sim hackathon of synthetic biology. And in this competition, we met Alonso and Grecia. So it's like a good bond between us. So I think it's that if my mates can like to.
And I think that they were an important part of the project because when we were into the bootcamp, it was like a lot of ideas of project that we wanted to create, and they were like the principal reason why we put the fits in the soil, and we were like, okay, we're gonna do that because it's a really project that can solve a problem in our country. So they help us to decide, for example, which one was a better decision, and they teach us a lot of things that we needed to know to be an IGEM. So I think that they were a really important part of the whole process to create this project, and they were like the principal part, because they were our teachers.
Thank you. Just so reminder that we cannot have any replies or comments from advisors. But we do still have some time for more questions from judges, so I will leave Margareta to ask her.
So I'm curious about the human practice part. Which part of your project within the human practice do you think was most successful, or which one are you most proud of?
Well, I think the part that was more successful was the scientific divulgation, because we get to go to stands in the university so the people can understand really the problematic, and also we make virtual meetings with girls that were really interested in synthetic biology, and also we comment about the problematic and the project. So yeah, I think the most important part of our project was the divulgation and also interviews that we did to these communities to really understand what they need. In addition, also we did a stand in our universities. In that stand, also we gave information about the project and what is biology, synthetic biology.
Thank you.
Thank you. We still have a few minutes if the judges have any more questions. Yes, Margareta, go ahead.
I'll throw in a short one. Did you encounter any ethical problems in this project?
Well, I think that one of the most important things that we take care of is the biosecurity using the killfish. and the other things. But also we have to see and make a research to the Cartagena Protocol. That is one of the laws that Costa Rica is subscribed to. So we have to very good reading and good search of what are the articles that we can just use in our favor, or we can avoid, like using directly the bacteria to the environment. Costa Rica doesn't allow this, so we took the opportunity to just use the enzyme in emulsion. So I think that is one of the most important things that we can think about.
Thank you.
Thank you. Okay, so if we don't have any more questions from the judges, then yes, okay. We will ask for the team members to leave the room. Thank you very much, everyone.
Thank you so much. Special greetings to Lucas and Papi.
Thank you.
So much.
Pura vida.
Pura vida
Thank
desde
you.
acá, Costa Rica.
Thank you.
Pura vida.
Thank you.
Bye.
Thanks.
Thank you.
We still have a few minutes to start the next session, so we can have another 3 or 4 minutes break. Thank you. Okay, we have, I think, a few of the team members in the waiting room, so I will start letting them in. Welcome team members from Cynthia team. General instructions and one reminder, if you have more team members joining, please remind them they need to add your team name so we can identify them and let them in. I will start by sharing your promotional video with the judges, and after they have watched the promotional video, we will start with the questions, and you will have from 20 to 22 minutes to reply the questions. So I will start the video now while we wait for the rest of the team members to join.
Porque nadie había comentado de su gravedad.
No creímos que fuera tan importante. Tumberjalata u ojo de poeta es una especie nativa del oriente africano.
Al grano, ¿qué está haciendo en Colombia?
Solo mírala, es hermosa. Fue introducida en América en el siglo XIX como planta ornamental. Mira sus bonitas flores naranjas.
¿Acaso están de su lado? Cubre bosques, devasta cultivos, compite ferozmente por recursos con las plantas nativas. ¿Qué la hace tan exitosa?
Una planta increíble, puede autopolinizarse. Su fruto revienta esparciendo sus semillas de 2 a 3 metros. Se reproduce con esquejes. Hay casi 1500 semillas por metro cuadrado, con germinación en 72 horas. Y las asociaciones micorrízicas, el clima y los insectos son sus aliados.
Distribución.
En Colombia se registra en todo el territorio. Nos preocupa el altiplano del oriente antioqueño, con observaciones entre los 1500 y los 2900 metros sobre el nivel del mar.
Solo en el oriente y en zonas tan altas. No es una planta nativa de zonas costeras.
Eso también nos preocupa. Se ha observado en ciudades más bajas como Medellín y Bello.
Quiero a todos fuera ya, solo requiero del personal científico. Aliados.
La alcaldía de Envigado y Cordales están trabajando sobre el tema. Tenemos aliados con el grupo de investigación de la Universidad Católica de Oriente, con los campesinos afectados, con escuelas y con la comunidad.
¿Y por qué no hemos podido con ella?
Una erradicación manual resulta ineficiente, el control químico peligroso y por control biológico se tiene conocimiento sobre la planta.
Esta vez perdimos, ¿verdad?
La biología sintética nos podría ayudar. Le presento Poetizar.
Los RNAs de interferencia son una herramienta de silenciamiento genético de organismos eucariotas como Tumberjalata, los cuales cuentan en su maquinaria celular con una serie de complejos proteicos que facilitan el ancla de dicho RNA a un RNA mensajero complementario, reprimiendo su traducción. Nuestra estrategia consta de la producción de escala de los RNAs de N. coli con el poder de silenciar la expresión de un gen de relevancia metabólica en ojo de poeta, logrando su aniquilación. Usaremos un mecanismo de expresión que facilite su distribución en nanopartículas que permitan su absorción en las células vegetales y conservar la integridad de los RNAs, garantizando selectividad, efectividad e inocuidad.
Okay, so we will start with the questions now. You will have 20 minutes. So if any of the judges have any questions that we can start with, please raise your hand. Yes, Telma, go ahead.
Hello, people. Congratulations for your project. Was very interesting. So my question is, you are trying to have this project that you're going to spray into the plants, and you promise it to be selective. How can you be sure that it will be selective if the plant grows everywhere and many other things alive will be in contact with your solution?
Hello, how are you? Do you hear me? Okay, so we are trying to guarantee this selective using a specific region of the DNA of the plant. And we are testing that with bioinformatics, such as alignment procedures, such as BLAST. But obviously we have to do more efforts in the future in order to ensure the selectiveness, because there need to be more sequencing efforts and investigation in order to make our product very specific and very safety when we apply it.
I would like to compliment my partner and my teammate. The thing is, at first in this design stage, in order to like guarantee the selectiveness, the thing that we did was first to look for like sequences specific for Thunberg Lara, and then we do like local alignment with BLAST, as you could read. Then if you could see like the BLAST, we chose some sequences that are only found in Thunberg Lara. Then we discussed like the possibility that we doesn't have like other matches because of the lack of sequences of other plants. Yeah, it's that there is a risk, but what we think is we have to do like a first design stage, but including the cost, obviously, we think that the for efforts for eradicate this plant is to do a sequencing. What's the thing about it? It has like a high cost. So if I don't know if you remember that in some parts of our proposal we had another proposal. The first proposal was with phenolacid and specific like pigment. I don't know if that's the word in English, like color, a molecule is specific for Thunberg Lara. And that could be a possibility to center in the pigments of molecules that are reported only in Thunberg Lara and like sequence them to guarantee that specificity.
Thank you. We have one question from Lucas now.
Yes. Hello, guys. ¿Qué más pues, parceros? I'm very happy with you, with your work. I saw in the modeling that you ignored the induction of the T7 promoter. So I want to know, why did you do that? What was the most important result in your modeling?
Okay. Hi, Lucas. It's a pleasure to have you here. Well, I think you're talking about we ignore even in the like modeling with we have like a simple model and a model that includes the induction, but we only use like the induction with IPTG that induces the T7 promoter, and not the induction with L-arabinose that what it makes is to induce the production of T7 polymerase. Why? Because in our build protocols, we use a concentration of L-arabinose that guarantees the maximum production, like maximum rate of production of T7, how do you say that, polymerase. But what we want to control, we have to have like a switch for security, even that we're not working with dangerous or hazardous molecules. And the IPTG regulation is like the switch we propose for the security. So that's why we simulated mathematically just with IPTG, because we suppose, or that was one of the assumptions we did, is that we have high concentrations L-arabinose, so the 70 polymerase is highly expressed, and if we want to control our circuit functioning, we will do it with IPTG that induces the T7 promoter under the lag operator, and yeah, that process that you could read. And I think, like you asked for like the mean results of our modeling, is that as Miguel said, there's few information about Thunberg Lara, and just having like some results of what could be the concentrations of the species that we're going to have is like a big advance, even though that we don't know like what's going to be like the functioning of the mechanism inside the plant. So as you could see, the concentrations, the rates of production of siRNAs is high. So if we have high like rates of production of siRNAs, it's more like probable to make it function inside the plant. That is like the things we don't know. Yeah.
Thank you.
Thank you.
Yes, Margareta, go ahead.
I was intrigued to hear that synthetic biology could be used to combat invasive plants. Could you mention any other examples where synthetic biology has been used to combat invasive species?
Hello, Margarita. Um, this technology that we are using with synthetic biology is very new in the field of controlling invasive plant species. I don't know like a specific species, but I know that it has been used for insects that are problematic for some crops. But I think that this is very new. There is very few investigation around the invasive plant species, but I think it has a lot of future because it can ensure safety in the way of controlling by this bioherbicide, and it can also ensure selectiveness. So I think by doing this kind of efforts, there can be a lot more investigation and new products in this area.
Thank you.
Margarita, I think Miguel has a lot more to say about it, but he can tell that the strategy specifically of synthetic biology using RNA interference is broadly used, is new but broadly used for controlling other type of living beings, like fungus, for example. It's reported broadly how to control fungus, like invasive fungus plants, by the use of RNA interference. And you will say like other ways of using synthetic biology for controlling invasive plants. Other way I saw was not for controlling, but for use them. Like they can have another use, so we can take advantage of it through synthetic biology. What's the problem with our local problem? Thunbergia lata is not a good plant in any way for our ecosystems. It literally makes our I don't know how to say that word in English. If you could help us, oh God. Chunk.
Or drown.
Like take the air of the other species, the nutrients. It like forms underground webs. It's too dangerous to say. We would use it with synthetic biology in other form. That's why we said we have to eradicate it, because like giving the community the opportunity to use it is to say them, please continue spreading this plant. So we have to eradicate, and we have found in RNA interference a big opportunity, a big chance.
If I can say something more, I just remember that, for example, here in Colombia, there is also another problem that can be solved using this technology, like the Fusarium oxysporum problem in the banana plants here. So that kinds of problems can be solved using this synthetic biology strategy.
Thank you again.
Thelma?
And on the same topic, is there any like national regulation about it, even when it's not a native plant from Colombia? You are disappearing biodiversity somehow. So is there a regulation about it?
Hi, Thelma. We try to control that and watch that regulation through the Protocol of Cartagena, that is an international regulation. And we also watched if in our university we have the correct laboratory levels to control that plant, and we had that was level 1 or 2. And also in our country we have some entities that regulates our problem that is called CORNARE, and it's an
environment regulation in our country, and we talked to them, and they say where our problem and our strategy was correct, and we have like the respect of them.
Here I would like to highlight one effort from Juliana Calderon and her team in human practices in approaches with CORNARE. It's the Environmental Authority of Eastern Antioquia. And we just not visited them, we talked to them about the problem, our solution, and they have gave us a letter. We couldn't send it to you because of the time they sent us, but they say we are one in this moment. If we start developing our project, we're one of the official, I don't know how to say that, I could show you the letter, efforts for eradicating the plant. And they have a lot they have a lot of strategies, important strategies with important research group, and it's like a big deal to be part of them.
Okay, thank you.
Thank you. Do we have any more questions from the judges? Yes, Margareta, go ahead.
So I'm curious about the carbon dot system. Could you explain or motivate your choice of the carbon dot system for distributing your system?
Yeah, so one of the problems that we have when we were thinking about our solution was how can we introduce the RNAs into the plant cells. So we found that there are some investigation around the use of nanoparticles such as carbon dots that facilitates the permeation of those interference RNA. And we also choose carbon dots because they are easy to produce. They are relatively cheap compared to other types of nanoparticles, and they also ensure a low toxicity and safety in the environment because when producing them, you use natural polymers or carbon compounds. So it is really like this safe. I think that it is a safe option that can ensure us high permeation rates and eventually high levels of silencing of the gene that we target.
Well motivated, thank you.
Thank you. We have one question from Lucas.
Yes, I saw in the collaboration. I thought that you talk with other universities. So in that sense of collaboration, in your job proposal, I thought several connections that you made. And I find the National Network of Synthetic Biology students in Colombia very interesting. So I want to know more about the network of collaboration. Can you tell me more about that, please?
Yes, thank you. First of all, yes, we did a lot of collaborations with several groups from Colombia. We wanted to talk with them because, well, this is our first time participating in the competition, as you may know. Yeah, we were a little bit inexperienced in some parts. We were asking like, what's happening here in Colombia? Who is talking about synthetic biology? What's going on? Why can't we find like more people talking about it? So yeah, we contacted the Theobroma IGEM team. That was the first one where we started like talking like, yeah, we should start like talking to others and to Colombia about this topic. Like we want all to know about it. It's pretty interesting. So yeah, with them we collaborated and we said like we're going to invite a few teams, not only from Colombia but from other countries, and we made a webinar. This was made with other groups, the National Network of Synthetic Biology. We explain our projects. We talk about how this synthetic biology could impact our lives and solve most of our problems like we were doing here. So yeah.
Thank you. Just to compliment, in Brazil we have a network like that. So do you think about making an association with this network, like to optimize the connection in the different place of Colombia, or now it's like a connection and network to talk about synthetic biology, like something more informal or something like that?
Well, it started like very informal, if you want to call it like that, because we have like a WhatsApp group and we're in contact. First of all, like speaking like, hi, we're from this group or we're from this university, because our partner, Theobroma, they've been like very active in contacting people who knows about this topic and are like here in Colombia, and they can introduce us to very other things. I think that we, with this group or with these students, we are going to do a few things in the future, to to share the knowledge. First, it's something basic. We're going to share the knowledge that we have, even if it's not very big, but we want people to be conscious of this topic.
Lucas,
Okay, thank you.
besides that, we have talked about like joining efforts not to make the like letting it die like a small I don't know, like a debate team with not that. We want to look for solutions not only for Colombia but Latin America. So we know, because of our collaboration, the big web of synthetic biology they have in network of synthetic biology they have in Mexico, for example. One of our biggest collaboration was with a team that was also working with an invasive plant, and we have talked about like making it formal and associated with other networks of other countries, because we have so much potential. And I don't know, with the knowledge of like expertising of Mexico, the problems and like the potential we have in Colombia, we could make great things, make great things.
Okay, thank you. Good job, guys.
Thank you. We have one question from Margarita.
I am just curious, which part of your project or your proposal do you think would be most challenging when it comes to do it experimentally?
In my opinion, I think perhaps the testing of the solution, because in order like to control every aspect of the project, we have to test like the formulations in real plants. So that can be a little bit difficult because sometimes you cannot control like the growing of the plant in laboratory. So I think that could be a little bit challenging, but I think with collaborative effort we can do it.
I agree with Miguel, but for other reason, because I think not because it would be like difficult like to test the function of the system, but because I think the most challenging like, step would be to make it real. Like, but the part of spraying, it's like broadly reported in literature, but in lab, yeah, we have nanoparticles are going to enter the cell. It's easy, relatively easy in the lab, but you know we have big webs of Convergallata along the country, webs, literally webs, and at first it could be easy in the lab to make it and prove it. Yeah, it's function, but when we go to the field, like make it. There are a lot of plants, there are a lot of environmental factors, there are a lot of other plants like making it difficult for the way of the nanoparticles. If they enter other plants, there's no problem, there's no security problem because of their selectiveness, but they're not gonna go to the target. I think it's like like the time when we're proving it, like testing it, but in field, not in lab.
If I may follow up. So if you specify the molecular part, which part of that do you think would be most challenging to do experimentally in the lab? Because I do agree, distributing the molecules will definitely be a challenge. But if I specify the lab part, what part do you think would be most difficult?
Maybe like the part of conjugating the RNAs with the carbon dots, because like those are nanoparticles, and I think it could be difficult to ensure that the bonding between those molecules is going to be 100% effective. Or also like the production of the RNAs, because those are very little molecules that can be easily degraded. So that is also a challenge that we have to be very cautious, and we have to have really good procedures in order to make a good experimental test.
Yeah, like I say, I think it's the same answer, both of them, because they're part of the same process. We have to like produce the siRNAs. It's easy, yeah, it's relatively easy. Spraying them are easy, but like we have it in the protein, in P19 protein, we have a protocol for, how do you say, isolate them, like, yeah, to isolate them, isolate them, yeah. And in the protocols, you can see we have like RNases inhibitors in order to guarantee the, like, I don't know how to speak English, like, yeah, but like the RNAs to be good, to be well. But you know, there are like 22 nucleotides RNAs, and they are small, they are little. But I think that with the protocols, like we could do a good work in order to like standardize a protocol that maintains like the integrity of the RNAs. But I think that's the like most difficult part, like isolate them and conjugate it with the nanoparticles. But it's something we could fix.
You're optimistic. That's good.
Thank you, everyone. So that ends our judging session for today. So thank you to everyone, team members, advisors, mentors, and judges for joining today. We will be finishing the judging session now. I will ask the team members to please leave the room, and I will stop the recording. Thank you.
Thank you very much.
Thank you very much.
Thank you. Bye.
Thank you.
Thank
bye.
you.
Thank
Bye
you.
bye.
Bye. Good luck.